Good. Nice, first one. Well, there was an intro, but don't worry. Hello and welcome to the John Spinach Start Week 2024 conference. This is Contributed Oral Presentations, Mobilizing Data theme, and I am your moderator, Javier Molina. Today, Jennifer Trimble is helping me here to fill any questions online. So I think by now you know the drill. Basically, during the session, any questions that you have, if you are online, just drop it out on Zoom. After the session, there is a chat on Oxford Abstracts to follow up the conversation. So without any further ado, basically I want to welcome here to the lectern Shelley James for the first session.
Thank you. It's great to see you all in here. Pack it in. Come on, pack it in. So yes, my name is Shelley James. I'm with the Western Australian Herbarium, and I'm here to present some work that was done with the Cambridge University Herbarium, and all my co-authors are either based there now or have since moved on. But, talking about mobilising international Australian collections locally for global benefit. So from the earliest days of colonisation and exploration in Australia, beginning with William Dampier in 1699 in Western Australia, to Banks and Solander on James Cook's first voyage around 1770, and then the first inland expeditions, botanical collections from Australia have been sent to herbaria overseas for Western scientific study. So within Australia, the earliest herbarium established was the National Herbarium Victoria in 1853, so reasonably young. For Western Australia, we're even younger. We have botanical collections retained in the state that date back to only 1896. So where there's a duplicate set of collections overseas, they may have been repatriated back to Australia with time, but many specimens, and particularly unicates, they remain housed overseas, and they're a bit tricky for us to get to in Australia. So these specimens document the floral diversity of Australian ecoregions that have now been altered, and of species that have not yet been described, and may have now been lost from our ecosystems. Many such collections are type specimens, lots of Australian type specimens overseas, and others have historical exploration significance, and most of them, these specimens are, as I said, not readily available for local taxonomic and cultural research without costly travel or the risk of shipment. One such herbarium housing early collections is the Cambridge University Herbarium, which you can see here. It's the fourth largest herbarium in the United Kingdom. It has the acronym CGE, or the Botanical Institution Code CGE, and it has a wealth of specimens from Australia. And I was very fortunate to receive funding from the Winston Churchill Memorial Trust in Australia to undertake the project I'm about to talk to you about. The project has enabled Australian collections to be located in CGE, updated, digitised, mobilised and shared. So it's decolonisation, colonisation and repatriation in digital form from Cambridge. So in June this year, just June, seems like just yesterday, I travelled to Cambridge to spend 5 weeks in the collections. With such limited time, the digitisation effort focused on the Australian vascular plant collections, and these were very fortunately systematically located in the world collections and geographically arranged, so they were all within Australian folders. But the collections of John Lindley were not so conveniently arranged, just by genus, species, and from taxonomic understanding of the 1800s. Awesome. So the already separated type collection was not a target for the project. Curation was limited to significant repairs and the incorporation of loose specimens into the collection. So now all Australian material has been pulled from storage and is ready for both physical and digital curation. The type specimens that were identified within the main collection were put into separate folders. You can see in the background on the stacks there, a bit of red, that's all the type specimens that were found. And barcodes were adhered to each sheet. Basic metadata, including taxonomic name on the sheet, storage location, country, and type status were gathered prior to imaging. We then imaged the sheets using two stations that you can see in the bottom image there. There's Ella and Sally, busy, hard at work. They couldn't keep up with me. They were rocking it. With two Nikon D850s, which are 50 megapixel cameras, DSLRs in light boxes. The raw files were converted to 16-bit TIFF for archival storage. They generated full-size JPEGs and 2 megabyte JPEGs for online delivery. So at this point in time, I've only managed to transcribe 5,700 of the collection items, and so the information that I'm going to be sharing with you is based on those specimens at this point in time for the most part. So what have we found? So within 5 weeks, more than 14,000 specimens were digitised. There was more than 13,700 collections from Australia. Some New Zealand, Pacific and horticultural specimens were housed with the Australian material, so they also got digitised. So there's about 500, 600, 700 odd specimens that, from other areas that have also been now released from the vault. More than 470 type specimens were flagged, and there were so many more that I actually had to stop looking because it was taking me so much time to write the determination and verification slips. 145 vascular plant families, about 875 genera, and 4,500 species. Many of those are still very, very old names. There's been a lot of work done since the 1800s on the taxonomy of Australian plants. And so I'm going through and now updating all of those names before we can release this data. So just to give you an idea of the scope of the collection for Australia, most of the collections were from Tasmania or Western Australia, where I'm from, so my government department was very happy about that. It advances our knowledge. But about 30% of the collections were actually just flagged simply as New Holland, which will require a little bit more research for us to be able to narrow down where they were collected. They were probably mostly East Coast, Victoria and New South Wales sort of areas. So within the collection there were some prominent collections themselves. John Stevens Henslow was the 4th Professor of Botany in Cambridge beginning in 1825. And he received many specimens as a result of his connections to other members of the British scientific community of the age. So that's how a lot of these collections are from Australia. And Charles Edward Moss similarly was a curator in 1907. He gathered a lot of materials from overseas. John Lindley, the Lindley collections were purchased in 1866 by Cambridge. They came from London and they're filthy. They are very, very dirty specimens because of the industrial age of the time, covered in soot and dirt, but they are fantastic collections. And so, yeah, they were purchased by Cambridge, except for the orchids, which all went to Kew. And then there's the Charles Morgan Lemon Herbarium. This is an example specimen from that. That was donated in 1852 after it went to Bennett for study. So, yeah, the Kew numbers are linked to wiki data pages for those people. So that's one of the things I'm madly doing, is adding everyone to Wikidata. Woo! So the top 10 collectors, Ronald Gunn collected mostly in Tasmania, James Drummond collected in Western Australia, Salisbury, Lister and Hearn collected in Australia in the late 1800s and early 1900s. And these collections are not represented at all in Australia. From what I can tell. So brand new material that we can have a look at. The other expeditions that took place, Major Mitchell, Thomas Mitchell, Alan Cunningham, Joseph Hooker, some material from Antarctica from him, and the islands just off Antarctica, Samuel Mossman in New South Wales. And one of 3 women, Georgiana Malloy, made the top 10, which is awesome. She collected in a vast area of Western Australia, down south. The collections mostly date, well, 1839 is very prominent there. That was Drummond collecting in Western Australia, a lot of that, and also gun collecting in that time period. But you can see all the collections from before we had herbaria ended up over in Cambridge and various other institutions. And then we have that spike in the early 1900s, with some collectors depositing their specimens in Cambridge. Oops, I'm already out of time. So what are the next steps? This project and the generation of so many images forced Cambridge to find a new data storage solution. No more hard drives, thank goodness. And all of their images are now actually online if you know the Cambridge barcode number. I'm madly working to complete transcription. The nomenclature and taxonomy is also being updated, and once that's complete, all of the images and data will be shared through the Atlas of Living Australia. And I'm hoping that in using the annotation tools of the ALA, the Australasian community of botanists, historians and the public will help to curate and improve the data, provide identifications for specimens and help to identify types and collectors from handwriting on the labels. Data, of course, will be fed back to Cambridge, and the data will be shared through GBIF, through Cambridge and other biodiversity data aggregators. Access to these specimens is already being requested. It's high demand, so in this age of accelerated biodiversity loss, and so we're working as fast as we can. And with that, I'd like to thank my co-authors for their assistance and enthusiasm. They worked like devils while I was there. Thanks to Sam Brockington and Ma for their support and the much needed help from many volunteers that helped out. Thanks to Tadwig and Spinach for getting me here and the Winston Churchill Fellow program. And finally, this project was considered as a seed project for accelerating the digitisation of Australian international material. And so if you hold Australian collections but do not have the resources to digitise them, I'm keen to connect with you to see how I can find some funding to get an Australian over to help you out. So, thank you.
There you go. Thanks, Shelley. Unfortunately, I think we have run out of time. So if you have any questions from Shelley, well, you know where to find her here. And also, if you are online, just jump to Oxford Abstracts, so Shelley can follow up any questions. So now let's move to the next presenter, please. Her name is Shao. Can you come to the stage? Sorry, to the stage, please. Thank you.
Good afternoon, everyone, and konnichiwa. So my name is Shao. I'm from Seattle, Washington. As the developer and the founder of eHerbarium, it is my greatest privilege to have the opportunity to introduce eHerbarium to you. So as plant lovers, botanists, or researchers, we all understand the importance of preserving plant species and sharing our knowledge. And eHerbarium provides a digital solution to do just that. So to begin, let's talk about herbarium. I should use the mic, right? To begin, let's talk about herbarium. Herbarium have played a critical role in plant identification, taxonomy, and biodiversity studies. It serves as a reference point for botanical studies. So traditional herbarium, however, has the limitation of accessibility and ease of use, especially in a world that is becoming increasingly digital. Just as Shelley has just presented before, in recent years, digitizing efforts are going on in many institutions, such as renowned institutions like Kew Gardens and the Royal Botanic Garden. So this is the slide, and the Royal Botanic Garden. Despite all these efforts, however, there is still significant needs for platforms that can centralize and simplify the access for this wealth of information, while also enabling collaboration and sharing. So this is where eHerbarium comes in. eHerbarium is a digital platform that integrates with the latest mobile, cloud, and AI technology, as well as the traditional herbarium and taxonomy insights. So it provides an easy-to-use user interface to access vast collection of herbarium images. In addition to herbarium images, it also supports live plant images. So whether you are a botanist, a researcher, or just a plant enthusiast, eHerbarium makes it very easy for you to explore, document, and share your plant information globally. So to start, let's have a quick video of eHerbarium. Hope you enjoyed the video. So here are some of the key features of eHerbarium. First, eHerbarium provide access to the vast collection of and high-quality herbarium images from Kew's Herbarium, the Royal Botanic Garden, one of the most respected botanical institutions in the world. The platform is extensible, allowing you to host images from other organizations as well. Second, in addition, users can upload, share, license their own images, or download images from others. They can also like any of the images. So additionally, the plant AI-powered tools help simplify the plant identification process. It offers auto-completion on taxonomy details and automatically automatically retrieving the corresponding scientific name or common name. Moreover, the platform automatically associates the image location to its level 2 TDWG region. These regions typically correspond to large country or subnational areas, help ensure the consistency and accuracy of the geographical referencing, while also respect the image owner's location privacy. Just for your reference, here is a quick overview of the TDWG level 2 regions and with a QR code for the original GitHub doc for your reference. So the most powerful aspect of eHerbarium is its ability to foster a global community. Users can contribute to the growing database. They can share their knowledge and finding with the world. This collaborative approach not only helps enhance the biodiversity, collective knowledge of biodiversity, but also help ensure science more accessible to the general public. So for organizations and researchers, eHerbarium serves as a centralized and easy to access database for research in the areas such as ecology, conservation, and taxonomy. The platform is extensible and customizable, so it can be customized for your each individual needs. For educators and students, the platform is a they can use it as a learning tool. So they can explore the plant kingdom in a more interactive and engaging way. Even the general public can benefit, gaining deeper appreciation of the world around us and of the plants around us and the world. So, since its launch in March this year, eHerbarium has gained over 40,000 downloads worldwide. Here are some of the image uploads from the users around the world. These images are from sub-Indian continents, Southern Europe, Eastern Europe, Northern Africa, and Northeastern USA. So, looking forward, we'll continue to expand our user base, and we'll also looking for any collaboration opportunities with other organizations, and any funding opportunities are welcome as well. In conclusion, eHerbarium provides a platform that for the herbarium and plants and the live plant images, provide a platform that live plant images. It's modernizing how we document and share plant information, and by leveraging the latest digital technology, it empowers the global community to contribute to the knowledge and preservation of the plant kingdom. So we would like to encourage everyone to explore eHerbarium. Let us know your feedback and join us in celebrating this incredible diversity of the world around us. And that's it. And hope you enjoyed it. Now I would like to, this is acknowledgement. The Q Herbarium data are queried from POWO, which is provided by the Q's Herbarium. And this project is funded by Pacific Technology and Resources, which is a company in Seattle, Washington.
Thank you for your talk. I'm wondering why, for live images, for example, you're only recording them to the TADWIG Level 2 standard. Just to say something comes from Australia is not a lot of value in that. When you compare to other databases of live images like iNaturalist and some of the other databases which record the actual locality with an uncertainty, and if you want to cover privacy, you can block that uncertainty down. So I'm wondering why you're using the live images and not just concentrating on the herbarium photographs, etc., the images from the herbaria, when there are other software that may do that, the live images in a more accurate way.
Yeah, so you have two questions. One is why it's using the level 2 images, and also why the software shouldn't just concentrate to herbarium images instead of also expanding to live images. I think, for first question, I think it's, good suggestion. The original consideration is a lot of software using the coordinates, GPS coordinates, which could be have the potential of interfere not with the user's privacy. So many of the herbarium images from Q's Herbarium, they use the level 2 geographical tagging. That's why we use that just for consistency with the Q's Herbarium data. I think moving forward, we may, if there's a need, we may try to providing more detailed, more narrowing down the geographical referencing for these, could narrowing down the geographical referencing for these images. That's a good suggestion. And the second question is why why we expanded to live images. I think the platform is just expandable. You know, it's we can easily include live images and Herbarium images, and there is we have different menus, so you can just choose for Herbarium images, and you can choose to see other user uploaded images. So yeah, it's expandable, and we could have an app just for Herbarium images, or could have a different profile. Just say if you just want to see the Herbarium, the app will just show you the Herbarium images. Yeah.
Thank you for your talk. It was very, very interesting. I was just wondering, you named the funder of the project, but it doesn't. I have no idea what this company is. Neither do I know about what is SAM registered US company. Oh. And my question is, maybe you could elaborate a little more on the funder and tell us if there is a guarantee that in 10 years this is probably a private enterprise, if they're not bored by the project and they just give it up and the funding is not there anymore.
Yeah, the company it's a technology company in Seattle. It's a registered U.S. company. It doesn't have our outside funding yet. It's just funded by the company itself. So that make it have the autonomy of implementing the features as we like. The SAM, it stands for SAM. It's I forgot exact, but it's a U.S. government. It's one of the U.S. government. SAM.org is, if you can just try. I don't remember the full name. SAM. It's a company for if you are a small business, you register with SAM, you can receive government funding. Yeah, thank you. Thank you very much.
Thank you, Shao, for your presentation. So moving on, let's go to our next presenter, Pei-Yu Tsai. Hopefully I pronounced it correctly. So over to you. Thank you.
皆様、こんにちは。私はTsaiです。Good afternoon, everyone. I'm Pei-Yu Tsai from Biodiversity Research Center, Academia Sinica, Taiwan. Today, I would like to introduce our recent work on spatial, temporal, and environmental completeness of eBird data in Taiwan. eBird is global citizen science programs. It's online, open access data, allow bird watchers to upload and share their bird species sighting in real time. Data correlation covers large spatial and temporal scale, and is well sampled by eBird participants with over 1.3 billion bird records on global scale. eBird provides a semi-structure that the dataset include that coordinates, timestamp, and survey protocol type variables. And eBird is utilized to study bird ecology and conservation policies. The application include bird attitudinal migration behavior and species population trends, migratory community monitoring, environmental interaction, such as wave turbine, site selection policies. However, eBird data presents a scientific challenge due to its opportunistic data correlation methods, which limit its application in certain research contexts. For instance, this study is used eBird global scale to analyze species distribution and assess data completeness. The researchers, they found geographic in data coverage, with well survey coverage in northern country and India. They also identify data gap in South Hemisphere country and North Asia. Due to this, bird research is still limited by uneven survey and uncertain biases in eBird data. Our next focus is on Taiwan. Understanding its unique location and its significance of bird conservation is crucial. With hosting over 600 bird species, Taiwan is a signal bird biodiversity hotspot for conservation. Situated in Southeast Asia, it's a crucial stopover and a wintering site for migration birds traveling between East Asia and Australia. Taiwan even now has over 11 million records, as the illustrate by the data increase over time. In the previous global scale study, Taiwan completeness seems enough, but if we want to examine finer spatial and temporal scale, is Taiwan eBird data still appear complete? It's crucial to assess the data completeness before any analysis. So in this study, our research objectives including assess eBird data completeness across spatial and temporal scale in Taiwan. Second is evaluate the environmental representation of the area with the high completeness of eBird data. We use Zhao diversity index calculation to calculation data completeness, which consider rare and unseasoned species in the sample, and allow estimate the total species richness in the specific area. We use INES, which is based on Zhao index calculations. Data completeness across sampling site can be calculated. This figure shows the species accumulation curve. The x-axis representing number of the sampling units, y representing number of the species. Data completeness is estimated by total number of survey species divided by estimated number of expanding species. In this study process, we first Taiwan was divided into equal area of each grid cells, and the eBird checklist data were mapping to these grids, and data completeness calculated using INEX package in R. Especially, we analyzed the change of the data completeness at 1, 5, 10 kilometer scale over 14 years. Temporarily, we assess the change of the data completeness every 10 years. In analyze, we define the well survey grids as the data completeness over 80%. Chi-square testers were performed to compare the distribution of the well survey grids to all grid surveys. The environmental factors include elevation, forest coverage, annual temperature, annual mean temperature, and annual precipitation. This figure shows data completeness of the grids at various spatial scale. Different color indicate data completeness value of each grid. Right grid cells represent well surveyed grids. We can see the data completeness improves as the spatial scale increase, with significant in ratio of the well surveyed grids across Taiwan. Hence, especially eBird data at 5 kilometer scale show the over 60 completeness is good enough for further analysis. We then use 5 kilometer scale to temporal analyze. We found an potential improvement of data completeness over time. The Taiwan mapper shows the data completeness of grids every 10 years over time. Temporarily, we can see the data in the last decade had achieved similar completeness to entire 4-decade data, indicating citizen science participation in the last decade has been very high. We compare the frequency distribution difference between all grids to well-surveyed grids. The blue colors indicates distribution of the all grids, and the red color indicates well-surveyed grids. The distribution of the well-surveyed grids is similar to all grids in terms of the annual presentation. It shows good survey coverage. On the other hand, we found data gap at high elevation and high forage coverage, and the area with lower annual mean temperature. The table shows the chi-square results for well-surveyed grids. Non-significant value indicator representative sampling of the environmental factors, such as forest at 10 kilometer and annual precipitation at the 5 and 10 kilometer scales. As the spatial scale increases, we can see the left figures, forest coverage and the annual precipitation achieve a higher sampling representation. In final discussion and conclusion, with our data found data gap in the area with high altitude and higher forest coverage region, and the place with lower average annual temperature that show where survey was still insufficient. And when use eBird data for ecological study, balanced sampling should be considered. We found some geographic bias in the well-surveyed grids. Balanced sampling improvements accuracy and application for ecological analysis and conservation planning. Representative sampling across spatial scale to determine the optimal resolution to capture organism and environmental interaction. In our data, we found eBird data in Taiwan is particularly useful, with forest coverage in the well represent at the 10 kilometer spatial scale, and the annual presentation is well reflected at the 10 kilo and the thinner 5 kilometer. Thank you for your attention.
Thank you. That was a very interesting talk. I'm wondering if you've mapped those areas where there's underrepresentation, the high mountain, the forest, etc., and made those available. Bird people are always up for a challenge, and if you put that out and say, these are areas we need more data, I'm sure they'll be out there within weeks looking for those. I'm just wondering whether you've done any work like that to try and bring the citizen scientists in to look at those areas.
I think it's all about access, right? The areas that are getting birded are the areas of the roads. Those areas.
Yeah, the bird people go anywhere.
Bird people are nuts.
If you're on a short trip and you go to the easy places there.
Yeah, that's true.
Do you the question?
Yes, in Taiwan, we do some benefit for actually we try to fill the gap, data gap at a 5 kilometer scale, because 5 kilometer scale is more suitable for Taiwan environmental, because the mountain area is the clear data gap, the site, yeah. Actually, we encourage volunteer to do that. Yes.
Any other questions? All right. Nothing else. Let's welcome our last presenter, Johanna Loacker. Hopefully I pronounced it correctly. Yes.
Well, thank you for sticking around and not starting your tea break early. My name is Johanna Loacker, and I'm representing the invertebrate zoology collection staff at the California Academy of Sciences. And today I'll be discussing how we implemented and refined direct and indirect transcription workflows as part of a grant period, and hopefully our experiences can kind of guide your upcoming digitization processes. So as one of 19 US institutions involved in the documenting marine biodiversity through digitization of invertebrate collections grant, we'll call it DIGIN for short, we contributed to building a new support network for for invertebrate collections. This network has facilitated the broader access of invertebrate specimen data, and as well as the sharing of knowledge and resources between invertebrate collections. We benefited greatly from LA County's workflows, and I know they're going to be talking about that later. So highly recommend going to see that. So our collections at the Academy, where our target was 60,000 curated non-molluscan marine records. Mollusks make up approximately half of our targets or our holdings, so this target is a sizable portion of our collections. And just to note that our invertebrate collection does not include insect taxa. So the extremely broad taxonomic scope of this grant, as well as a heterogeneous collection in various states of curation, made our workflows difficult to plan. And our small but very mighty team on this slide, this is the extent of our invertebrate collection staff. We have 3 permanent staff, and then Hannah Beck was our full-time digitization technician position that was funded for our grants. If you do the math, all 4 of us can't digitize 15,000 records each, so we needed to be creative in how we used our resources in getting to our goal. Our standard direct data entry workflow mirrors the approach that we used before the grant period, with one exception that transcribers now enter data directly into Excel sheets instead of entering data directly into the database. This is beneficial as permanent staff can now review and quality control data before it is uploaded. And it also restricts direct editing rights in our database to prevent miscellaneous deletions or errors. It's also a little bit more accessible for new transcribers to enter data into a familiar space like Excel versus specify forms. I briefly mentioned our transcription rate. That is with our DIGIN technician as well as some volunteers. I'll talk about that a little bit more later at the end of the presentation. And to just briefly summarize our label transcription workflow, we created a Scrappy, so to speak, a resourceful scanning workflow with 2 main goals. Efficiently using volunteer hours to support our DIGIN technician and generate label scan images that can be used for offsite and direct transcription. So as I said, to briefly summarize, I'll get into a little bit more detail later. Scanning in orange and label transcription in green. We initially tested a combined workflow with volunteers where they did scanning of labels as well as skeletal data entry, but that proved to be inefficient as volunteers struggled with the dual tasks. Separating scanning and transcription allowed volunteers to focus on a singular task, which boosted efficiency and satisfaction. So for scanning specimen labels, we had 10 volunteers during the grant period that created label scan images in 2 separate fluid collection rooms and to avoid moving specimens between the floors of our building. We also had a dry scanning station set up in an office just outside of our dry collections room. Each workstation featured a Caesar document scanner, and in our wet collection we used laptops for ease of use and mobility. Placeholder jars, which unfortunately aren't in this folder or these images, but are bright orange, made reshelving more accurate and quickly for our volunteers. And scanning time varied. Quicker for section with fewer labels and longer for lots with vials, which took a lot of time to remove the tiny labels out for more legible scans. And I don't have time to go in the depths of our label scanning workflow, but you can always come talk to me or contact me later. So after scanning, image files and Excel sheets were sent to both volunteers and our transcriber when she was working from home. This remote transcription was advantageous, especially for a remote volunteer who appreciated the flexibility to contribute to our project at her schedule and her pace. Since volunteers weren't physically handling specimens, keeping them engaged by other means was crucial for retention. Volunteer survey feedback indicated that support, education, and a positive attitude from their supervisor was crucial for their satisfaction. So, however, managing volunteers had challenges, particularly with asynchronous error correction. Label images did help permanent staff quality control and spot errors, and these label images will be included in the collection object record and specify for future condition reporting and error checking later on. But despite volunteers handling most label scan transcriptions, significant staff time was needed for training, pre-populating data sheets, correcting errors, image file management, and uploading data into Specify. And so this slide is a brief and small and not exhaustive selection of our various types of labels that we have both in our fluid and dry collection. And our label scanning workflow was highly iterative. When transcribers faced issues or uncertainties during the transcription process, we adjusted our label scanning workflows accordingly. And I'll just share 2 of the adjustments that we've done to deal with difficult specimen data. So to maximize speed when transcribing from our labels, transcribers did not research station or expeditionary data that was not easily accessible in our database. For labels such as this one, where just a station number is present, transcribers would use this text string in the orange box here to indicate that no further locality is found at this moment. While this choice allowed for transcribers to move more rapidly, it is generating a significant amount of staff time in the future. To produce complete records, we will need to research data on this station. We also had a separate text string for when there was no locality present at all, and that is also indicated in the locality name of our collection event record. And I would say the most significant challenge we had was dealing with specimens whose determinations did not match where they were shelved in our collection, or who had several determinations listed on labels. For label transcribers, this was problematic as they did not have the context of shelving order from just looking at a scan. To address this, we began including the taxonomic label from either the shelf in the dry collection, or the wet collection, or the drawer label in the dry collection. And this can be seen in the bottom part of the scan. We call them context labels. There's number of specimens, preservative, and then that determination, which often aren't present on our historic labels. Unique identifiers were assigned by scanners as well, and the QR code is embedded with a unique identifier that helped us speed up our image file renaming process. So transcribers would enter the determination from this context label into the shelved as field in their spreadsheets, which populated a text field in our specified collection object record. Printing and stuffing labels was not included in our grant workflow to maximize time spent generating records. This field will record where the specimen was last found and hopefully aid in future label stuffing and reorganization efforts. You may notice this specimen does fall outside of the scope of our grants. When we finished scanning the grant-related specimens in our collection, we utilized our volunteer workflows to scan selected areas of our mollusk collection. And so this chart summarizes our progress since the grant began. Our grant was awarded during the pandemic. So as soon as we were able to access our collections, we needed to hit the ground running. Scanners started label scanning in fall of 2021, and our our dig-in technician was hired in February of 2022. This image does highlight the work of our one grant digitizer, Hannah Beck, represented in blue. She's amazing. We're very lucky to have her. And she was a former volunteer in our collection. And so hiring volunteers is beneficial as training time is reduced. And they can just start as soon as possible, and it is great to pay our people who work there. And transcription weights did fluctuate over time and gradually decreased as we reserved the more complicated section like formalin preserved specimens for the later periods of our grants. And here are some of our totals and rates for our 2 transcription workflows. The 40,000 records produced during these 2 workflows will supplement a third volunteer-based workflow on Zooniverse that unfortunately I'm running out of time and will not be able to talk about today. But our findings indicated that for our collections, direct data entry was significantly faster, nearly doubling the rate of label transcription workflow. As I'll discuss, only looking at digitization rates can be misleading and not the only factor when determining a workflow for your collections. So comparing transcription rates can be challenging, as previously digitized data was spread across our collection, changing the amount of data that needed to be entered. While our volunteers transcribed faster when working directly from specimens, the workflow that they were doing often only included entering the collection object data. Variations in transcription speed were influenced by the complexity of taxa and the curation needs, and this can lead to discrepancies that don't fully reflect process efficiency. Lastly, digitization rates overlook the essential contributions of permanent staff. And I'm running out of time, so I won't be able to go over all of these considerations. Oh, cool, well then I have time. Sorry, everyone. So as many collections push to digitize their specimen data and seek external funding to support their staff, hopefully some of these questions can guide your planning. Each collection is different, and each workflow should be different depending on the needs of your collection and those who use them. Let's see. So first thing, and I think what is most important, is to define your data needs early, so you can direct your approach and your allocation of resources. While curatorial staff love to capture as much data as possible, maybe you can consider producing a higher volume of lower quality records during the grant period, with more detailed work to be done later when you actually know what you have in your collections. Consider whether your focus should be broad or a specific taxonomic or geographic area. And so for us, the both direct and indirect workflows demanded substantial staff time for pre-curation, management, engagement, and quality control. This time, although hard to quantify, is significant. So plan for more staff time than anticipated, and know that even like cutting labels takes a lot of time. And our volunteers contribute over 3,000 hours to pre-curation, scanning, and transcription. While volunteer voice workflows are valuable, they should not replace full paid time positions on future grants. Ensuring the equitable museum field means not creating barriers for those who cannot work for free. And lastly, being resourceful or scrappy means just leveraging external support networks and internal resources to achieve your goals. If you don't have an external support network, maybe you can create one. And you can achieve goals when your resources are limited. And I wanted to thank our Dig-In community for supporting us, and hopefully we supported you slightly. And lastly, our Dig-In volunteers, that we would not have been able to accomplish our work without them. With that, I will take any questions.
I was wondering if you've looked at the Digivol process that's been developed for using volunteers for transcribe the data.
And so, as I briefly mentioned, we had a Zooniverse transcription project that kind of ran simultaneously with these two workflows. And I don't know how much I can delve into that, but we had thousands of external volunteers that transcribed our catalog cards that were scanned during the pandemic. And that also required a substantial amount of staff time. I scanned all 40,000 cards. We had to answer all the questions on Zooniverse. And then that data that we received from Zooniverse is a bit messy, and so we were actively cleaning it up as we speak. So that will eventually get pushed, as with all this data that we produced, into our Specify portal and other aggregators. But it's a great resource to use.
So can you just confirm, I don't know enough about Specify. Does Specify allow you to add an identifier for the collector?
For the collector,
Yeah.
I believe you can have it, put the ORCID ID on
Okay,
there
just
as
to
well.
let you know, your first slide had a Thomas A. Birch. I've now just put him in Wikidata, so he's got an identifier for you if you need it.
That will be another project in the future for us, so thank you. And if you think of them later, I'm around. Oh, absolutely, I will stay right here.
Okay, so I think it is very interesting, basically all the workflow that you are describing. I was wondering whether the project that you are running has any room to look at automatization. So basically, because I see some of those processes can be very repetitive, and I can see, well, indeed, scanning just herbarium sheets, there are a lot of projects basically just looking at, I don't want to call AI, but just automatic image identification. precisely to speed up the process.
And so we do have some labels that are typed out on these little 3x5 cards. I tried to use OCR very briefly in the card catalog scanning process, and it was awful, at least with the resources that we had available. As you can see on this slide, we have things that are handwritten, things are attached to specimens, things are written on the specimens. So it's just a great variety of things in the invertebrate collection that make it very difficult to have one exact workflow. It'd be amazing if we could do something like scanning herbaria sheets. It's just not quite feasible, especially for things in our wet collection. But it's something we can hope for. Thank you, though.
Any last questions before we wrap up? Thanks, Johanna. Well, then, this brings us to the end of this session. So, Nife Debiru, thank you, everybody. So, you're welcome to join the afternoon team. Thank you, guys.